Now I'd like to introduce our speaker. Sarah Laske is a fish biologist at the U.S. Geological Survey Alaska Science Center in Anchorage. She was hired in 2017 after completing her PhD at the University of Alaska Fairbanks, where she studied drivers of fish community structure and trophic relationships in Arctic coastal plain lakes. Her work focuses largely on fish community ecology and food webs of Arctic species and landscapes. Currently, she is taking on investigations exploring mercury biomagnification in lake food webs. Outside of work, Sarah is at the whim of her spoiled bird dogs, but she does enjoy baking loaves of sourdough and other treats. Welcome, Sarah Laske. At this point, I'll turn the webinar over to you.
Hi, everyone. Thank you for coming. We just have to get things rolling here. Okay, can you guys see the screen?
Yes, we can see the screen.
Perfect. Okay, there were a few debacles this morning before all of you joined. Anyways, thank you very much for coming. I'm excited to share this work that we did a few years ago and it's just finally wrapped up into conclusion in the earlier part of this year, assessing freshwater biodiversity. And today I'm just going to talk to you about the fish component of that and highlight some of the environmental drivers and composition of fish species across the circumpolar Arctic. Okay, so the Conservation of Arctic Flora and Fauna, CAF, is a working group of the Arctic Council that established the Circumpolar Biodiversity Monitoring Program in order to address the needs of coordinated monitoring on a circumarctic scale. The Freshwater Group developed a monitoring plan and compiled and analyzed the first ever circumpolar database of freshwater organisms to assess status and trends in Arctic freshwater biodiversity. It's important, first and foremost, that I thank the individuals from those organizations and this slew of co-authors here for their work that they performed getting these analyses ready. All of these co-authors that I've mentioned here were on two of the papers that I'm going to highlight in today's presentation. So to begin, at large spatial scales, either continents or regions, historic and modern features of climate, geology, and hydrology often influence species distributions. Temperature and precipitation patterns and events like past glaciations and the location of glacial refugia can all influence current and regional pools of species that occur in an area. Separations of species by habitat type, whether or not it's in a lake or a stream, or whether or not it's a large river or a small creek, can influence which species are present and the overall diversity of a system. Further, for anadromous species like a salmon, the proximity to the sea is important for their distribution and occupancy within a habitat. And as you move down in spatial scale from across a region into an individual lake or stream reach, the heterogeneity of individual habitat types within those systems may support or filter out different species. So we know from previous assessments and from just published literature that there are approximately 125 species of freshwater fish in the circumpolar Arctic, but little is known regarding the processes that drive their biodiversity in Arctic freshwaters. And this is surprising given how integral fish are to freshwater ecosystem function, and because they're often the top consumer in freshwater food webs and serve as a link between freshwater ecosystems and terrestrial ecosystems. Further, fish are an important subsistence species for most people who live in Arctic towns and villages. And so not only do fish play an important role ecologically, but also for the humans that rely on them. So understanding their distribution and diversity patterns is important for their conservation, management, and monitoring. So why do we care? Well, biodiversity is at risk, particularly from rapid changes in climate. There was a recent paper that came out that highlighted the fact that rather than twice the global average, it looks like the Arctic is actually warming 4 times global average. So you have this graphic here of the top of the world that shows both the temperature trend of at least 1 degree C warmer and a much higher amplification rate than was previously known. So this astounding rate of change that we can see on the landscape. So I have this background photo here of a bluff that's eroding away from the permafrost thaw. And this is actually on the coastline of Kaktovik, and just seawater erosion. So these changes in temperature and this warming is going to lead to an organism response that will be rapid. And we lack data, we lack current data and assessment of current patterns that we need in order to establish a baseline from which we can measure future change. So in addition to those direct effects of climate change, warmer climate can provide more opportunities for human interactions in Arctic habitats, which could increase fishing pressures or lead to introductions of southern species. You have like the signal crayfish that's now in parts of Alaska, and also will increase potentially the landscape change due to energy production, oil and gas development, mining, and the like. So collectively, these stressors have the potential to change current biodiversity through change in the number of species, composition of species, or the variation of species number or composition from location to location in the Arctic. So I'm just going to highlight a few studies that have been conducted recently that sort of talk about some of these changes. I apologize, this figure came out a little bit blurry, at least on my computer, so bear with me a little bit. But there was a study done in Iceland and Norway that looked at the composition of salmon, trout, brown trout, Arctic salmon, brown trout, and Arctic char in Iceland and Norway's streams. And so that's what this figure represents. So it's the proportion of fish, so 5%, the amount of each species of the total, over a 26-year record from the early 1990s to about 2017. And over that time period, mean annual temperatures increased 1.7 degrees Celsius. And what the authors of the study found was that there was an increase in the proportion of brown trout and a general decrease in the proportion of Arctic char. And what they suspect is happening is that the slightly warmer water favoring brown trout will replace Arctic char over time. Additionally, authors from a paper in Sweden and Finland found that in lake systems, that the fish density and biomass increased with temperature and productivity. So the further south you went, the more fish there were, but they're smaller bodied and they're more warm temperature tolerant. And then as you move north, you get more large bodied cold water species. So you have this gradient of Atlantic salmon, perch, and minnows from north to south. And what they were discussing in their paper is this potential for a northward boreal shift. So you'd get more of these minnows and perch moving into systems further north as those systems become more productive and as they warm. And lastly, there's this potential for greater colonization. So one of our co-authors on these papers, Martin Svenning, put out a paper in 2015 that highlighted the first incidence of three-spined stickleback in Svalbard. So Svalbard is a remote island in the Norwegian Sea. It's located about 78 degrees north latitude. The only fish species prior to these potential colonizations of three-spined stickleback was the Arctic char. So basically the warming of waters in the marine environment made it easier for these fish to move from the coasts of Norway and Finland offshore and onto Svalbard. So this would essentially double the number of fish species on the island of Svalbard if those fish can successfully colonize. So the objectives of this work broadly, because I'm going to cover a couple of different studies, were to gather and harmonize available freshwater data. Also to determine multi-scale patterns of diversity. This includes alpha, beta, and gamma diversity, which we'll go into the details of those in a little bit, and also determine the processes that underlie fish community diversity. So first, we had to gather all of this data, and this was quite a large undertaking. It involved individuals from 8 different Arctic nations, and it was collecting and harmonizing data from monitoring, government reports, academic research, and industry. And what this left us with was like a variety of different types of data. I have this table here that I pulled out of the State of the Arctic Freshwater Biodiversity Report that highlights the different types of data that were potentially available to us. And in the end, when you account for the different types of gear and the different studies that were conducted, each with their own disparate needs for things that they were trying to attain, you end up with incidence data. So you have just slews of presence absence data and then pockets of other things that might be useful for case studies. So we pulled out those incidence data in order to assess an entire community, and then used data only from gears that were less selective or were used in a group of gear types in order to assure that. Any repeated sampling, so you went to the same lake a few times, we just collapsed that and reported how many fish occurred there, and then just used presence absence data for the full analysis. We also analyzed all of these presence absence data, diversity data over various spatial scales, the lowest of which was the hydro basin. So if you imagine the watershed, within that you have the various sampling stations where you collected the fish. So this is our smallest scale. The next scale that we looked at was that of the ecoregion. So I'll orient you here to this map. Again, this is the Circumpolar Arctic. You have Greenland here, Canada, Alaska, Russia, and Fennoscandia. So each one of these ecoregions is highlighted in a different color. So each one sort of represents natural communities that share a majority of species or environmental conditions. And so we used any of those that overlapped within the Arctic boundary. The last grouping was the zone, the Arctic zone. So again, similar orientation of the map. And these are shown in different shades of purple from sub, low, and high Arctic. And these were defined based on largely on vegetation and climate. So as I said earlier, I'd get into these diversity measures, the first one of which is alpha diversity. This is generally recorded at the smallest scale and is just a count of the number of different species that are present. So if we considered that for these little circles here, for each, we'll call them lakes just for, that's what they look like. For each one of these, we have these cartoon fish in, right? So it's just simply the number of unique species within each one of those systems. And this is a scale that's common to research studies. Often we sample a stream reach or we sample a lake, and that's the number of fish species that are present in that system. Alpha diversity can also be used for determining whether or not there's a diversity hotspot. So is there some place where you went and sampled where there was just a much higher number of fish species than anywhere else that you went? So second is gamma diversity, and this is the number of species in a region. And this is most often the scale that is used by society for maintaining biodiversity. So you think about, oh, the number of fish species that live within your state. And so this is often where decisions are made in the scale that we rely on. And that is basically, what is the total number of all species that occur in all systems? And so here, gamma would equal 5. So that's the total number of unique individuals. Now, linking these two things together is beta diversity. And this is the final measure that we're going to use in these analyses. And that is the dissimilarity among locations. And so how does the variation change in species composition and number from place to place to place? And this heterogeneity of species can occur across environmental gradients. So again, we have our little cartoon lakes. As the lake gets smaller, you lose species. And this loss like this that I've shown here in this graphic is a loss in an ordered manner, and that is what is considered nestedness. And so this is one component of total beta diversity, is this species dissimilarity via loss. Another possibility is a change, like a complete switch in the types of species that you have from one location to another. This dissimilarity is change from replacement or turnover. And when you have just all of your change due to that, turnover will make up all of your beta diversity. And we often see this in species compositional changes across biogeographic provinces. Or one example I thought of would be to compare the species assemblage you have in the Mississippi River to the species assemblage you have in the Colorado River. They're completely different, right? So these two things combined, that diversity due to species loss and the diversity that might not be due to losses in species, but changes in species, make up the total beta diversity. And this link here, the beta diversity link, provides a way of integrating diversity across spatial scales. So our data distribution, this is what we came up with after many hours of compiling and organizing data. We had 3,148 stations around the circumpolar Arctic. Again, I'll orient you to the map. We have Alaska here, across Canada, Greenland, Iceland, Fennoscandia, and far western Russia. And what you can see is we do have this quite sizable gap across most of Russia where we have no data. So we're really just working within this 234 degrees of longitude. We covered 4 zones because we added a few boreal sites down below the subarctic. This covered 25 ecoregions and 299 hydrobasins. We also had the majority of our sites were found in streams, 67% to 33% in lakes. But that's just like a side note that doesn't come into play in any of the analyses that I'm going to show you here today. But one other really important thing that I want you to take note of is that in this, I said there are about 125 species overall. So automatically we're missing 25 species from this region here. Within these 234 degrees of longitude, there are 100 known species of fish from literature and expert knowledge on just what occurs in those ecoregions, but we only had 56 in the database. So we're already missing about half of the species that we know can occur in these regions. So that's just something to keep in mind, that, you know, try as you might, it's not often perfect. Okay, so we found that alpha diversity, so again, that's that lowest level of diversity. It's the number of species found in a location. So in our instance, we used the hydrobasin. Found that alpha diversity is related to latitude across the circumpolar Arctic. And below 70 degrees of latitude, we had about 1.3 additional species over those 10 degrees from 60 to 70. So we're actually increasing in species richness. Now, one little caveat here, we have really low species richness around 60 degrees of north latitude. That's also where Iceland is, and Iceland is relatively species poor. Then when we get above 70 degrees, we have this gap where we really don't have any sites. And then there's just one species above 70.7, and that's the Arctic charr. And we'll come back to some discussions on Arctic charr in a bit, but it's really interesting that you have just this fairly dramatic decline in species richness. So diversity varied among our zones and ecoregions. We're talking now about gamma diversity. This is the much larger scale. So this is the number of species across an ecoregion. And we found, if you look at this map here, I've highlighted it, you have sort of at your lower latitudes here in Fennoscandia and Russia, we had 47 unique species. We had 26 unique species in the coastal tundra of Alaska. But then when you get into higher latitudinal zones and further north ecoregions, you have 2 species. And like I said before, when we got up really far north, we only had 1 species of fish, the Arctic charr, that was a record. That's because you need to be both a hardy species and have that ability to swim through salt water. So you've got to leave the mainland and get out into this archipelago. So we really lose continuity of habitat when you get that far north. Also isolation, like I said, if you have to traverse the sea, you have to be a fish species that's adapted to do that. So even at high and low latitudes, we lost species richness if you're in an isolated system. In Iceland, there's a total of 8. All of these fish have some anadromous capability. And then in Svalbard, it's generally Arctic charr, but we have that possibility of those three-spine stickleback moving into those systems. So again, you see this decline with north across zones, and you have a reduction in richness or the number of species there with isolation. So we wanted to break this down and look at the composition of individual species across each one of these zones. So across the bottom here, moving from Alaska through Canada, through the North Atlantic and into Scandinavia and Russia. And this is a breakdown. And if you want to see, there is a full table that complements this in the paper that was in Freshwater Biology. I'll get to some details on that at the end of the presentation. But basically, each species is lined up along the y-axis here. And the large takeaway I want you to note is that these are the species that are represented here. There's a trickle that go across the middle, and then these are the species that are represented in Scandinavia and Russia. So we see that full-fledged turnover of species. It's a different set of individuals that occur in those different locations. So of the 100 known species that I talked about in the previous slide, 79 turned over across continents, with 29 occurring only in North America and 50 occurring only in Fennoscandia and Western Russia. 17 species occurred across the continents, and 77 occur in the high and low Arctic zones, but 23, mostly minnows or cyprinids, which are represented by these blue dots, they occur only in subarctic and boreal zones. So when we think about the different species that are occurring here, we have our circumpolar species, so Arctic charr, the stickleback, those occur everywhere. Then in Fennoscandia and Russia, minnows and perch are very common and unique to the, and not necessarily unique, but just more abundant. But then as far as endemic or unique to North America, you have suckers, which we don't have in any other parts of the Arctic. So when we considered alpha diversity as this related to various environmental factors, there are a couple of things that we found. And we plucked out data from those places where we had a lot of points. So locations in Alaska, Iceland had good, robust data. And then in Scandinavia, we had robust data. So we pulled those out and then we rarefied those values so that we've leveled the playing field essentially. So we said, regardless of how many sample stations you collected from, we're stopping at a certain point. This is how many species we have there. And so we've taken that into consideration. And so first of all, in Iceland, remember that was isolated in the Northern Atlantic. There were only 3 fish species represented in those samples there. So that isolation really kept it from having higher diversity. When we look at these other regions, I've overlaid these maps here, you can see that there are low elevation sites and high elevation sites in both of these larger regions. And in both cases, where there are mountains, there are fewer species, right? So that was a significant difference here in the number of species between the Scandinavian montane birch forests and the Russian taiga, right? And the same thing in Alaska, coastal tundra had more species than the Brooks Range did. One thing to note here too is that in these coastal systems, you get a little bit of species richness bump. The number of species is a little higher because you have these coastal or anadromous species that you don't have inland. So you have both of these things sort of at play. And so that richness that you get at higher latitudes, we saw this reflected just in this separate study that Jennifer Lento led, that looked just at watersheds in North America. So this is just in Western North America. And we looked again at the richness of the hydrobasins, and we had again this peak in richness in Northern Alaska here. And this is related to the glaciation history of the region. So the coastal plain here was in a glacial refugia during the last ice age. We found here and in Europe that those glacial refugia are really important to predicting diversity patterns now. If you were a refugia then, you have more species now. So when we wanted to look at the community composition that's associated with this biogeography, we did this again in North America. We looked at Canadian sites, which are these blue triangles, and Alaska, which are the orange. And we plot those out in ordination space, each point representing a basin community. So this is all of the species collapsed into one point that occur in one location. And we can see that the spread of Alaskan sites, the variability amongst Alaskan sites is much greater than the variability in those Canadian sites. And when we consider the fish species, these are the open dots here with the abbreviations, these are the species codes. When we consider which species overlay on this and which species are driving these separations, one of the most interesting things that, that I noticed is that you have this separation between very freshwater species or slightly warmer water species like pike in Canada. And then in Alaska, you have the whitefish, the blackfish, stickleback, things that are cold water tolerant, or even have a little bit more tolerance to salinity because they live along those coastal habitats. So coming back around to beta diversity, so this was the variation in composition or richness from site to site to site. We looked at this first across our Arctic zones, and we found that there's a general decline in beta diversity the higher in the Arctic you go. So when we look at the component parts of beta diversity, the nestedness and the turnover, we find that turnover or species replacement was often the dominant component. And this is likely due in part to the large spatial extent, particularly in the boreal zone and the low Arctic zone. In the boreal zone, there was a maximum linear distance of about 6,400 kilometers between sites in Canada and Russia that we were comparing. So biogeographically, we know that there is going to be different pools of species in those continents. In the high Arctic, if you look at the median of both turnover and nestedness, those are very small, but beta diversity is relatively low there. And there are fewer species in that region. And so when we dig into it, we look at each region, we found first that there's really no variation in our data set in the high Arctic ecoregions. So nestedness was equal to turnover, which was equal to 0. And that's because in our data set, we had one fish species occurring there, and that was Arctic char. So when you move from site to site to site across the high Arctic, you just caught Arctic char, caught Arctic char, caught Arctic char. And this isn't to say that there is no diversity. There certainly is, but it's within species. So it's intra-species diversity instead of, across species. So that's just like a whole other set. There are a lot of interested researchers who are digging into this. It's certainly a hot topic for char and whitefish. So that moving forward, there'll be a lot more work done there. If you consider the ecoregions where we sampled and had a lot of robust data and also covered fairly large spatial extents with that data, species turnover was dominant in those areas. So the nestedness or the diversity due to loss was outweighed by the diversity changes due to composition. So then in other locations like Iceland, again, you have this location that's relatively isolated. You have fewer number of species there to begin with. The differences among watersheds there were driven by nestedness. So it was an ordered loss of species within those systems. So in an isolated system like Iceland, any change in your community is going to did result from losses across a richness gradient rather than a swapping of species. And then lastly, all these purple shaded ecoregions, we found that there really wasn't a dominant driver of the beta diversity. It was in equal parts nestedness and turnover. And this is a combination of both losses of species richness and changes in species composition. And one of the things that's important to note here is that in these systems, we really didn't have a lot of data. So it could be a function of just poor data numbers, or it could be a function of something that's truly happening. Certainly, there is no reason why both of these components couldn't be contributing to losses, but it's certainly more dependent upon spatial scale. So to wrap up all of those things, all of those analyses that we did from those two papers, the diversity that we found was correlated with environmental factors, generally latitude, spatial isolation. So you think about Iceland or the Canadian archipelago. The glacial history, was there a refugia where fish species were protected during the last glaciation event? And then also coarse ecoregion characteristics, such as was it high elevation or not? So all of these things combine together to create the communities that occur in those systems. And the number of and composition of fish species depends on their dispersal abilities and also on historic and contemporary barriers to their colonization, which can influence local and regional diversity patterns. So determining dissimilarity among communities, including differentiating turnover and nestedness components of total diversity, is critical for understanding ecological and conservation issues. The use of beta diversity along with species richness studies allows for scaling up local studies to inform regional conservation decisions and is essential for analysis of mechanisms and processes that underlie biodiversity. It's essential when considering the proper monitoring protocol, whether that be the total richness in an area or the average number of species, interest in heterogeneity among systems, or even composition of species richness along an environmental gradient, be that a stream length or elevational gradient. So in addition to everything I shared with you today, there are a number of papers that we put out in part of a special issue in Freshwater Biology, Ecological Change in Arctic Freshwaters. There are 11 other papers other than the two I highlighted today that are all open access, so they're freely available for anyone to read. We discuss circumpolar assessments of freshwater chemistry, diatoms, zooplankton, and phytoplankton. There's studies on macroinvertebrates and also on indigenous knowledge. There's regional assessments, again, that cover ecosystem patterns in subarctic lakes, diatoms, macroinvertebrates and fish, and also plankton and myobenthic fauna. And then there's also one-time series in there. I did highlight some of that from the Freshwater Biodiversity Report on salmon, charr, and trout in Iceland and Norway. A more detailed view of even that work is the State of the Arctic Freshwater Biodiversity Report, which came out in 2019. This assesses drivers of change, the status and trends in biodiversity, shares a lot of the same information, but with greater detail that we shared in those papers from the special issue. It also includes a biodiversity synthesis and the state of monitoring in all of the Arctic nations, and offers up some recommendations for people moving forward in terms of improving monitoring or working to collect more data. So, like I mentioned, that State of the Arctic report, really the main thrust at the end of that was that gaps persist in our data, right? Across countries and habitats, here we've got lakes and streams across both habitats and organism types. So working down fish, invertebrates, benthic algae, the abiotic system, the consistency of monitoring is variable across countries, and it makes global assessments difficult. So each one of these colors represents a different level of monitoring. So the blue dots, like you see in Sweden and Finland, indicate that sites are routinely monitored with consistent funding and good spatial coverage. The majority of places have something that falls in between that. And very rarely there's absolutely no coverage, but it does occur. And so, in Finland and Sweden, standardized government monitoring programs provide data that's of strong temporal and spatial quality. And in other countries, we maybe rely on data that's not part of any monitoring network, but is instead based on irregular and secure funding. And so there are emerging approaches to future monitoring that include the use of remote sensing, environmental DNA, citizen science, and traditional knowledge that can all be used together to inform biodiversity studies. Moving forward, we're continuing some of this work, but we're looking at different levels of diversity now. So we're looking at species functional traits and also at interest-specific diversity, like I mentioned before with the charr. So future investigations will continue, and we're going to keep expanding our data sets to include forms of diversity other than species composition and richness. So at that, I'm happy to take any questions. I'm also going to point you to a couple of resources if you're interested. For more information just in general on freshwater biodiversity in the Circumpolar Arctic, and any data resources that are available from these studies can be found at the Arctic Biodiversity Data Service. So thank you very much, and I'm happy to entertain any questions.
Thank you, Sarah. That was really a good presentation. And as a quick note, today's presentation has been recorded and it will be archived to view at a later date at your convenience. We're now going to start the question and answer session. As a reminder, if you're joining by Zoom, please raise your hand if you'd like to do that, and I can call on you, can ask your question aloud, or if you'd like, you can type your question into the chat window and I will read the questions aloud. I don't believe, if you're joining by phone, please raise your hand by pressing *9 and I'll call on you, and then you can ask your question aloud. Then you'll need to unmute yourself by pressing *6. And if at that point, let's see, I see one question from Caitlin with the U.S. Coast Guard. Can you put those links into the chat? The links to the papers, I believe he's asking for. And I don't see.
I could copy them in.
Thank you.
Might take me a minute to find the chat, but I will do that.
Yeah. I do not see any hands up. Thank you, Sarah, for putting those up.
Oh, you bet.
I have a kind of a basic question, and that is about the refugia related to glaciation. Can you give a better, more of a definition of what that is and how that affects fish populations?
Certainly. So in the last ice age, there were pockets of the north that were ice-free. So water continued to flow, fresh water continued to flow, and fish that lived in those systems persisted through the ice age. They weren't squeezed out, they didn't perish. They just sort of consolidated into those locations, right? So then when the ice retreats, you're left with this little hot spot of diversity where then everything else gets recolonized from. And it's hard to move, right? It's hard to move away from a system. So you were consolidated in this location because it was ice-free and you could live your little fishy life without the risk of death. And then the ice moved and all of those fish had to move back out of those refugia. So it's true in, there's studies that have been done in Europe and in North America. There's a really good one that was put together, I think it's Oswood, I don't remember the year, for Alaska, that just talks about the several papers by Griffiths. If you're really interested in it, there's a lot of good resources out there. But basically it just talks about time since glaciation and how rich different areas are. So really having that safe space where there was no ice helped preserve fish species in higher numbers. And that carries through to today. There's like a legacy of that on the landscape.
Thanks.
Mm-hmm.
There's a question from Carl Rowland. Can you unmute yourself and ask your question, Carl?
Yeah, hi. Thanks for your talk. That was interesting. Yeah, and you were kind of getting at what I was going to ask in your last answer, but I was curious if you're aware of studies specifically of expansion, the rates of expansion into peripheral glaciated regions from these sort of refugial areas. Like, have people looked at the rate? I imagine it's variable, but I'm just curious of explicitly looking at the refugial areas and then how fish are dispersing into previously glaciated terrain.
Yeah,
Thanks.
that's a great question. I can't think of any off the top of my head. I've stumbled into quite a few papers over time about the exchange of fish in space. I can't think of any that specifically discuss rate, but that doesn't mean I haven't encountered it. It just means it didn't stick. But that's an excellent question. I certainly think that there's probably even new research on that happening now with glacial retreat and fish moving upstream. So you could probably even find studies, I would assume, that talk about movement upstream of melting glaciers at this point. So
yeah, thanks. One thing with plants is that you have these recontact areas where they were separated and they come back in contact, and you get creation of novel species from that contact. Of course, plants biologically very different. That's another thing I'm kind of curious about, maybe more phylogeographic.
Thank you. The next question is from Falk Houtman. Can you unmute yourself and ask your question? Then we have two
Yes.
questions in the chat.
Yes, hello. Can you hear me?
Yes.
yes,
Okay, cool. I had a question about human influence on the distribution. In some areas, there have been people living for thousands of years, and you haven't dealt with that. I think humans are probably have a big impact on wiping out or changing species compositions. I wonder what your comment is on this. Thank you so much.
yeah. We did catalog incidences of species introduction, and that's mentioned a little bit more in the report than in the papers. We just didn't have enough robust data to really get into that, like timing of introductions and that sort of thing. But we did catalog where species were introduced, some of the Pacific salmon in Europe, brown trout in the United States. So we did look at that, but that is a great point, particularly in regions that are more well populated. There's always that possibility for facilitation and movement of species across boundaries.
Yeah, just to comment there, what I was referring to is anthropogenic, like indigenous use of species, because there has been a long history of humans living in the subarctic, certainly in the Arctic too, and they modified the lakes and the waters. So it's not recent modification, really modification in the last 500 or 1,000, 2,000, 300, 3,000 years.
Yeah, that's a really interesting point. Unfortunately, it was kind of outside of the scope of what we did in this paper, but that's certainly an interesting topic and hopefully one that we can dig into. Thank you for the question.
The next question is from John Sonnen on chat. He says, thanks, Sarah, quite thorough, but on biodiversity interspecies, have you seen any changes yet due possibly to warming waters shifting northwards?
I haven't worked on any of that myself. I'm trying to think if I've stumbled across anything within species for diversity changes. There's a lot of work with collaborators in Norway and Finland looking at whitefish, but I can't think of anything off the top of my head that was specifically related to warming. It's more just a habitat use question. So I would have to do my homework to look into that a little bit more. I'm sorry, I don't have a better answer for you.
Thanks. The next question is from Tai Ding. I attended EUSPA Cassini competition last year about polar. People should include space data from sun enlargement, Earth move away from the sun, sunlight and moonlight intensity change, nuclear leak data, actually global cooling, ice ages bigger volume than water, sea level, and will still raise. I want to build a vegan robot to grow vegan and polar to serve people. Do you have any advice?
Not any advice specifically. I do know that there are, in terms of like different types of data, I know that there's been some work towards moving towards more remote sense data for this biodiversity work, particularly because that's something that you can deploy fairly easily and get large swaths of data that then, you know, there's less cost of having to put people out in the field. And when you do that, you only get like a small location, right? So I think there's some push to get more into that remote sensing world, where hopefully we can come up with some standardized metrics of things to measure, you know, chlorophyll in lakes or something like that, that could help us inform about biodiversity change over time.
Thanks. I have another question myself, and that is looking at the lack of data over such a significant part of the Panarctic over the Russian coast. Do you have any kind of idea of what type of data might be missing or how that could affect the biodiversity? Any guess? I know it's just a guess.
We are missing, I mean, so much data, like spatially from Canada, even we were missing large swaths of data. Our subarctic data was actually fairly limited compared to what you'd think you could have. And I think it really just comes down to, you know, like it's not, nothing is standardized and nothing's ever been done in a way that's, the end game is not, we're going to inform circumpolar biodiversity, right? Yeah. Each project has its own critical objectives that they're doing. And then as someone trying to gather that data, you have to contact each individual who's maybe been somewhere and collected it. So I think, I mean, I'm very curious to see how things would be different, like how much more diverse are things than what we could see in this report. I'm guessing quite a bit more diverse. You know, but it's just, it's a huge gap. It's an unfortunate circumstance, but one that we have no choice but to sort of find ways to deal with. And hopefully, as we move forward, we can come up with more collaborative ways of filling in these data gaps, that we can have more robust data to answer questions.
Thanks.
Mm-hmm.
I don't see any other questions or comments. So kind of a last call here for questions and comments for Sarah. It's been a really good presentation. I'm sure people will want to follow up with you. A couple more minutes here. Sarah, did you have any, were there any questions that you think folks might have, did you have any other comments to ask or to add? Any other?
I mean, I think this is all incredibly fascinating. I love it. I love beta diversity as a tool. I think it's something that's getting more popular as a way of thinking about diversity. And if anyone is really interested in it, there's some great R packages for it, and some great papers. Package beta part was what we used to do this analysis. It basically uses community matrices to look at changes or differences in composition. So that's just a little nugget there if anybody's trying to get into the beta diversity studies. There's a lot of great papers out there.
Thanks.
Mm-hmm.
Well, if there are no further questions or comments that are coming in, I think that we are ready to close. And on behalf of Arcus, I want to thank all of you for joining us today, and a special thanks, of course, to our speaker, Sarah Laske, for her excellent presentation. We are continually working to improve our seminar series, as well as looking for suggestions for future seminar topics and speakers. Please take a minute or two to complete our short survey. A link to that is on the screen and also available in the chat window. Today's webinar will be archived and available on the Arcus Seminar Series webpage. An email containing a link to that page will be sent once the video recording has been posted online. Please visit the Arcus homepage for more information about Arcus, our programs, and upcoming Arctic-related events. Thank you again for joining us today, and thank you especially to Sarah Laske. And I believe this concludes our webinar. I wish you all well and have a good day and a great weekend coming up. Thank you.